This week, we're going to look at mosquitoes, bedbugs, and lice. The learning objectives for this lecture are to identify mosquitoes, bedbugs, and lice based on their morphology and biting habits, their life cycles, their distribution, associated human diseases, signs of infestation, and control and prevention using integrated pest management strategies. At the end of this lecture, students will be able to describe the following human diseases transmitted by lice. That's typhus, based on clinical symptoms, epidemiology and transmission routes, laboratory diagnosis, treatment, control and prevention, and list other human diseases transmitted or caused by bedbugs and lice. Mosquitoes have similar life cycles to flies. They undergo complete metamorphosis. All types need standing or stagnant water to breed. In North America, they are considered a nuisance rather than pests because when they bite, intense itching can occur at the site. Only female mosquitoes bite to get the blood meal for egg development. There are two major groups of mosquitoes, the culocines and the anophelines. You can see that the culocine group has a larger genera of mosquitoes that transmit vector-borne diseases than the anopheline group. There are many different genera of mosquitoes, and it is difficult to differentiate them. It really needs some experience to look for variation in mouthparts and banding patterns on legs. However, this is important because different types of mosquitoes transmit different diseases. The Anopheles mosquito has different mouthparts to mosquitoes in the culocine group and spots on its wings, whereas the Aedes mosquito, which is a culocine mosquito, has prominent banding patterns on the legs. In Canada, Culex mosquitoes can not only transmit West Nile virus, but also viruses such as the EEE virus, that's the Eastern equine encephalitis virus, and the St. Louis encephalitis virus. The other diseases transmitted by mosquitoes are mainly found in the tropics. This is a summary of the major human diseases known to be transmitted by mosquitoes. It's quite a long list, and I won't read them all out. I have placed asterisks beside the ones that we'll be covering in this course. Most of them will be discussed in the vector-borne disease lecture at the end of the semester if we have enough time. As I mentioned before, the mosquito life cycle involves complete metamorphosis. When the mature female adult mosquito lays its eggs in stagnant water, they hatch in a couple of days to produce larvae that wriggle around or hang upside down, with their breathing tubes sticking up at the surface to take in oxygen from air. After a week or so, the mosquito larvae become pupae, and they stop feeding and become less active. Winged adult mosquitoes develop within the pupae and emerge after a few days. They then mature into full-grown adults to complete the life cycle. Aside from the morphological differences in their wings and mouthparts, anopheline mosquitoes can be distinguished from culocines by their resting positions and when they bite. Anopheles mosquitoes stay at an angle, whereas culocine mosquitoes lie flat. Culex mosquito eggs are found in rafts, whereas eggs from the Anopheles mosquito are separate. Let's examine the mosquito mouthparts more closely. Although both the anopheline and culocine female mosquitoes have long needle-shaped biting parts called the proboscis, they have a pair of palps on either side that are different in size. Anophelines have palps that are long as their proboscis, whereas the culocine palps are short, and only the antenna and the proboscis project prominently from the top of their heads. Therefore, to distinguish a female anopheline mosquito from a culocine, you should look for 5 long structures at the top of the head, whereas culocine mosquitoes only have 3 long structures and 2 much shorter ones. Male mosquitoes have different mouthparts to females because they don't bite to take blood meals. They are vegetarians and feed on fruits and plants. You won't need to differentiate between male and female mosquitoes for labs, but you can see that male mosquitoes have antennae that are covered in fine, feathery, hair-like structures. As part of the surveillance of West Nile virus and other mosquito-borne viruses, such as the EEE virus, public health units in Canada have carried out mosquito surveillance on a regular basis since 2002. The program enables the tracking of diseases that are transmitted by mosquitoes, as well as the monitoring of new mosquito species and genera in Canada. Mosquitoes are tracked and sent to the public health units for testing. Some of you might have participated in mosquito surveillance during the summer and seen these traps. They don't all look alike, but essentially they have a source of carbon dioxide, usually in the form of dry ice, as bait for mosquitoes. The mosquitoes are also attracted to light, and there is a battery-powered light bulb which has a sensor that automatically turns it on at dusk and off at dawn. Trapped mosquitoes are usually collected in the morning, then frozen to kill them before being shipped off to the labs for testing. The latest available complete dataset for 1 year is from 2018. If we take the total of all 4 provinces that carry out mosquito surveillance, 3% of the mosquito pools were positive for West Nile virus. Ontario usually submits the largest number of pools for testing compared to the other provinces. The datasets for 2019 and 2020 have yet to be compiled into final reports, but it appears that there were lower numbers of West Nile virus positive mosquitoes this year and last year. In 2018, as well as previous years, the majority of West Nile virus positive mosquito pools were located in the Golden Horseshoe area, southwestern and southeastern Ontario, predominantly in urban areas. This is because there are large numbers of catch basins with standing water that provide ideal breeding grounds for mosquitoes. West Nile virus was detected most frequently in 2 Culex species, that is, Culex pipiens and Culex restruens. There could be a bias, however, because these 2 species are the most frequently tested ones, since they are the primary vectors responsible for transmitting the disease to humans. This is a map of the location of the mosquito pools that were positive for West Nile virus. You can see that the hotspots are around the Golden Horseshoe area, and the highest numbers were submitted from Halton region. This has shifted from the past when Peel had the highest numbers. If we look at the seasonal variation as well as temporal variation, there is a trend that can be observed, with more positive mosquito pools in years that have warmer summers as well as winters. This is particularly striking in 2012. Temperature affects both mosquito development and viral replication in the mosquito. This means that warmer summers will result in more West Nile virus positive mosquitoes and ultimately human cases. Colder winters in the preceding year will result in fewer mosquitoes in the following year, as adult females are unable to survive the harsher winter conditions. In addition to the West Nile virus, mosquitoes are also tested for the triple E or Eastern equine encephalitis virus. In 2009, the triple E virus was reported for the first time in mosquito pools in the Simcoe-Muskoka area. All the mosquitoes were culecines, but they were different to the regular Culex pipiens, Culex restruens mosquitoes that carry West Nile virus. Instead, there were other types of mosquitoes that preferred to inhabit flooded forests and swamps that are found in the rural areas and not in urban areas. In the past years, there have only been 4 reports of mosquitoes that tested positive for the triple E virus. Controlling mosquitoes is obviously very important in public health. The best strategies are those used in integrated pest management. Since mosquitoes don't fly very long distances and stay close to their breeding sites, eliminating these should be the first step. All items that can collect water should be either thrown out or covered or inverted so that water cannot pool in them. This includes many items such as pet bowls and bird baths. Ornamental ponds and swimming pools need to be aerated so that the surface of the water is continuously agitated to prevent mosquitoes from laying their eggs. Gutter and eaves troughs should also be cleared. For exclusion, any holes in window screens and doors should be sealed. It's important to wear long sleeves and pants or protective clothing that will prevent mosquitoes from landing on bare skin and taking bites. They are also attracted to dark-colored clothing, so wear lighter colors. When sleeping, use mosquito nets to provide more protection. Insect repellents can also be sprayed on clothing and exposed skin. They should contain DEET, but at no more than 30% concentration for adults and 10% for children. DEET-based repellents should not be used on babies under the age of 6 months. And if you're only going outdoors for a short period of time, 30 minutes or less, then DEET may not be necessary. For agricultural and community control, biological control can be used. Fogging using a bacteria, BTI, that kills the larvae specifically has been very effective. Fish can also be useful in ornamental ponds as they will eat the larvae, but are obviously not desirable in swimming pools. Perhaps the most common form of control is using chemical control with methoprin, a chemical that kills larvae by mimicking the juvenile growth hormone of mosquitoes. It is used by many municipalities in the GTA to prevent larvae from developing in catch basins. Alternative chemicals are the pyrethrins from chrysanthemums and pyrethroids, which are the synthetic versions. There is also a chemical called malathion, which is used in agriculture, as it degrades rapidly, unlike older chemicals such as DDT. However, malathion is highly toxic to fish and honeybees, and is therefore not environmentally friendly. I've included this extra slide to show how Public Health Ontario tracks West Nile virus. I've also added a link to a video that shows you how to use the graph to display the datasets.